Okay, so the purpose of this video is to give you a little more guidance on how to complete the first part of this lab, which involves this PowerPoint that you will have downloaded. There are a couple of tasks that you need to complete in the PowerPoint. And the first one, part one here, is going to have you show that you know how to recognize what cells look like in the different stages of the cell cycle. These include interphase, prophase, metaphase, anaphase, and telophase. So for part one, the goal is to refer to the second slide on here, which contains this very high definition image of the tip of an onion root. And as you can see, there are little boxes in here, each representing a single cell. And inside many of the cells, you can see a darkened material, and that is the DNA. Now, in order to determine which stage each cell is in, you'll need to refer to page 3 of the lab instructions that are posted on Blackboard, because on that page, there is guidance and also example photos of what cells look like in each of the different stages. The goal is to find a cell, any cell, doesn't matter which one, that is in each of the 5 different stages listed here on the first slide, and move one of the color-coded boxes around that cell to show that you can recognize that stage. So the purple box needs to move to a cell that's an interphase. So my recommendation is that you grab the purple box, move it somewhere in the middle of our onion root tip image here, and then zoom in. And as you can see, as you zoom in, you can get a better picture of what each cell looks like. And interphase, cells that are in interphase look like they have just a solid dark blob inside of them. Mitosis has not yet started. So I'm going to pick this cell and I'm going to move my little box right here so that that cell is highlighted in the middle of the box. And that completes the task for interphase. Now for prophase, metaphase, anaphase, and telophase, you'll want to look around and find other cells that match the characteristics of what it looks like to be in that stage. But once you have moved all 5 of the boxes into this area here and matched them with an appropriate cell, then part 1 is complete. Part 2, the instructions for which are in slide 3, is also going to involve using this very same photo here. And the goal of part 2 is to look at 100 of the cells. There's many more than 100 in this picture, but you want to look at 100 cells and figure out what fraction of them are in each of the different stages. We're just using 100 cells as a representation of the bigger picture of what's going on here. You don't have to count every cell. You only have to pick 100, and any 100 will do. The thing is, though, that you want to make sure that you don't count the same cell twice. And so the easiest way to do this and ensure that you're not counting the same cell twice is to rely on the natural organization that's already existing in this root tip, which is, as you can see, the cells are divided into these rows. So if we start somewhere over here on the right-hand side, and it doesn't really matter exactly where you start, but if you follow my cursor, let's start right here. This cell is in interphase. So that's my first of the 100 cells that I'm going to count. On the side, I'll have a scratch paper, and on my scratch paper, I'll have 5 different categories. I'll have interphase, prophase, metaphase, anaphase, and telophase. And for each of those categories, as I go down this row, every time I see a cell, then I'm going to make a tally on my scratch paper indicating which category it's in. So if we go, okay, this one's in interphase, this one's in interphase, this one's in interphase. Now we should have 3 tallies for interphase. We keep going and we see the next one is, this one looks more like prophase, right? It's not a solid dark blob anymore. The chromosomes are starting to take shape. So now I put a tally under prophase. And now we keep going. Interphase, 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 interphase. If you get to a cell that you can't really tell what it is, you can just skip it because it doesn't really matter which 100 cells you pick. We're just trying to pick 100. And then you keep going down this row and keep following it, tallying as you go until, and this will take some time, but by the time you reach over here, you should be at close to 100 cells, if not at 100 cells. And you want to keep going until you reach 100 tallies. You don't have to count 100 of each cell at each stage, you just want 100 total cells. Once you have your 100 total tally marks, then we move to this table right here on slide 4. In this first top row, this is where you will put in the number of tally marks that you've recorded for each of the different stages. So let's just say, for example, you counted that 80 of the cells out of your 100 were in interphase. You'll do the same for each of the other stages here. How you complete the second row is we're going to make the assumption that if 80 out of the 100 cells were in interphase, that means that the cells spend about 80% of their time in interphase. So this second row is just a percentage number that matches whatever you came up with for the first row. The last row here involves taking your percentage number and using it to calculate how many total hours out of a 24-hour cycle the cell spends in each of these stages. So if the cell spends 80% of its time in interphase, then that means we need to figure out what's 80% of 24 hours in a day. So to do this, you would take 0.8 and you would multiply it by 24. And that will give you the number of hours that these cells spend in each stage. In this case, if you had 80 as your number, it would be 19.2. Now, in all likelihood, you're not going to have 80 as your number. It's going to be however many cells that you count. But this is how you complete the table and do the calculation for each of the rows here, each of the columns.